we'll be discussing the RDCA-DAP data interrogator, how to sort, find, and integrate data, and how to develop an analysis dataset. Please keep yourself muted for the entire webinar. We will have a Q&A at the end of this session, so please put your question in the little Q&A chat box, and we will bring your questions up to our presenters at the end of their talk. So without further ado, it's my pleasure to introduce you to the Critical Path Institute Data Collaboration Center team. We got Vicki Crider, Senior Project Manager, Amanda Borens, Executive Director for DCC, and Kurt Michels, Data Analyst. Next slide.
Thanks, Alex. I'm advancing slides. I've got a little bit of delay. Guys, I'll go ahead and take the baton here. Get that slide moved forward. Thank you, Alex, for that wonderful introduction and getting us started and kicked off today. What we're going to talk about is our RDCA-DAP, and we'll talk about what that is and what it means and how we've gone on this journey with this particular solution. But I want to take a step back as the project manager for the development of this platform and tell you a little bit about the why and how we got here. And focused in rare disease, the rare disease arena, there's tremendous opportunity. Over 350 million people in the world have a rare disease, which doesn't in and of itself sound particularly rare. But when you look at the fact that that is split over 7,000 different options or possibilities for diseases, that really narrows or thins the numbers a little bit. And so drug development, as a result of that, is often slow because of low patient population or limited understanding of disease progression. And as a result of that, then you don't see a lot of investment. Not a lot of clinical trials go into that, and it just becomes a sort of vicious cycle for that. The United States enacted the Orphan Drug Act a while back. Other countries have similar laws. And so even with those efforts, it has created very little intangible results for that. Only about 10% of rare diseases have seen FDA-approved treatments based on the efforts of these additional laws incentivized to get drug companies to invest more in rare diseases. So with that tremendous challenge comes the opportunity piece and what we can do to help fill a gap in that environment. And that's where RDCA-DAP comes in. It stands for Rare Disease Cures Accelerator. And as any legitimate tool, it has an acronym. Ours is RDCA-DAP. It's a mouthful, so nobody would say that all the time. But it does stand for Rare Disease Cures Accelerator Data and Analytics Platform. It's an initiative funded by the FDA. And with our partners at NORD, the National Organization for Rare Disorders, we have kicked this off and stood up a platform to fill this gap. We are working with a company called Iridia. They are the vendor for this particular platform, and it lives within their digital research environment. Our initial hub was set up within Amsterdam, which gives us the flexibility to work with data owners and contributors around the world. And when we have such little amount of data available, that really gives us more opportunity to bring data in and make it available. So what really is RDCA-DAP? I want to talk a minute about that because this is the end where we're going today. So we're going to start at the end and then work our way back up to it. RDCA-DAP is an integrated data platform, an integrated database, and an analytics hub that's used to build novel tools to accelerate drug development across rare diseases. And I want to talk just a minute about that statement, across rare diseases, because we talked about the challenges when there are not many patients available within specific rare disease diagnoses. And with that comes limited data, and doing research on those limited data doesn't often give a lot of insight because there's just a little of it there, and what there is is often protected behind a veil because of security concerns and things along those lines. And so if you're looking just at Friedreich's data, Duchenne's data, maybe GNE myopathy, there are very small amounts of those data available. But if you could take those out of their silo and you could look at all of those data together, what kinds of additional little golden nuggets could be found? That's what we want to accomplish with RDCA-DAP. We want to make those data available for research and analysis across rare diseases. The DAP promotes the sharing of critically important data across those diseases for the specific purpose of accelerating the understanding of disease progression. One of the challenges, one of the limitations that is currently seen in the rare disease arena, and then optimize clinical trial design. So now instead of a vicious cycle, you have a positive loop where we're learning more and then feeding that back into the system and moving knowledge and understanding around these diseases forward. On the screen, what you've got is an overview of kind of how the platform works. We're going to do just a high-level discussion of that. We're bringing in data from various owners and locations and various types. And so we had to change that paradigm at c-path a little bit and expand what we're able to work with and accommodate for. Clinical trial data, registry data, and natural history data are already living there in the platform. We're working with those data now. We're in the process of also onboarding EHR data, which will be helpful. And then genomic and imaging data are on the roadmap for the fall of this year and starting to implement and integrate those data as well. We bring all of those data in to a common location, a common source, and they go through a process for curation, of course. But because these data are so different and variant in the types of data, the formats in which we receive them, we had to have a better way to really work with those data. So we're implementing ontologies and establishing mapping for these data on a high-level automated basis, which gives us some amount of standardization, and we can do some cleaning and formatting really easily because of that ontology level being implemented. And then as a result of that, we receive data that's available in the platform in a few different curated levels. We have a full curated, minimally curated. I anticipate we may even see some more options available there as we go. And then what becomes available from all of that work with the data to make it accessible within the platform is within the platform, you can then use basic data integration tools to understand those data better, to know what fields will be in a particular data set, or even what values are contained within those fields. If you want to know, for example, a dosage field, you can look to see what are all of the different options for dosage that I would see if I wanted to work within these data. That's available. Out-of-the-box analytics and visualization tools are also native within this platform. We're implementing more as well as we receive feedback and continue on this journey to determine what's helpful and valuable for researchers. You have the ability to develop analysis subsets of patient-level data to really get a deeper sense of what is in those data and what it could be used for, what they could be used for. That's being seen in a new cohort builder that we're developing right now. It kicked off a few months ago, and we're excited to see the opportunities that this is providing for our platform as well. And then advanced analytics capabilities exist within this platform as well for really deep understanding, deep analysis of the data to facilitate development of disease progression models, clinical trial simulation tools, and feeding into that positive loop that we talked about in that conversion. But how did we get there with those pieces that are available within the platform? I want to take a step back to walk through the beginning. So I told you now we go back to the beginning, and I want to take you through a journey of a proof of concept that we did within c-path to get the type of learning that informed these particular features. So we're going to look at our approach with a proof of concept, which was a data interrogator that we developed, share the functionality that was developed within it, and then the insights that we gained specifically around data exploration and visualization and how helpful that can be and what pieces make the biggest difference. And then we'll look at how those insights informed the roadmap, the development plan for RDCA-DAP, like we talked about. So our proof of concept, we developed a data interrogator, and the purpose of that was to identify our initial use cases in this project and really determine how we needed to solve problems and which specific problems were the best to solve in the world of rare disease and really offer something spectacular. We were able to align our existing experience at c-path in data curation and standardization and offering those data to researchers and working with them on that level. And then ingest also some new learnings and turn those into the requirements used to build the foundation of the platform and what was needed when we went looking for a particular vendor. It allowed us to experiment with new workflows and technologies and tools and determine what are ways this can be done better, what's possible, what's out there that we maybe haven't explored yet. And then we turned those into, like I said, the insights that really informed the development of RDCA-DAP. And we also spoke a little bit about that cohort builder that we're just now in development on. It really, that was one of the biggest pieces, one of the biggest golden nuggets, you could say, that we learned from the data interrogator and knowing what types of things needed to be built. The interrogator was an internal product that we developed using R Shiny, hosted within our own cloud environment. Initially, we set this up for Friedreich's ataxia, but we expanded that after we got some of the basics taken care of to allow for other diseases and to pull them out of those silos that we talked about. So you're not just looking at Friedreich's or Gianni myopathy or Duchenne, but we can look at all of them and we can facilitate exploration across those barriers. Visualizations, we say that a lot, but really one of the things we learned was how important being able to easily visualize data up front in the beginning makes the journey for the researcher, really facilitates that understanding. Metadata views are a piece that we learned were crucial as we moved our sites toward building and implementing changes within the development or within the data and analytics platform. Catalog entries for each data set was important to be able to facilitate doing that within the interrogator. And so we translated that also into the analytics platform. And beyond catalog entries, detailed data dictionaries for each value within those data sets, each field and what's included in those really opens the door to being able to utilize those data early in a more powerful way. Basic analysis at an early stage and being able to run simple analytics and statistical tests against data sets that are being identified within this tool were really helpful and really gave us a lot of information in informing where we were going with the overall platform. So that's a little bit of history on how this particular piece of our exploration or discovery phase of the project really informed a lot of the very important pieces that have been implemented and are continuing to be developed within RDCA-DAP. I'm going to turn it over to Amanda and Kurt because they're going to give us a detailed level or a detailed view of what I've just been talking about. This high level information that I shared, they're now going to show us what that looks like. Before I pass the baton over to you, Amanda, I just want to remind everyone that we will do questions and answers at the end, and we're excited to see your questions. We want to have that opportunity with you. Please make sure that as we're going through, if you have any questions, put those in the chat. We can discuss those at the end. And with that, I think I'll go ahead and turn it over to you, Amanda, if you're ready to pick up the ball there. I'll take us to the next slide.
Yeah. Hi, everyone. My name is Amanda Borens. We had a really quick introduction, and I've actually met probably some of our participants here, but Kurt might be new to you, so I'm going to take a moment to describe our relationship with Vicki. Vicki is our project manager and runs our operations branch of the Data Collaboration Center. I lead the data science team and have a background in data science. Kurt, on the other hand, has a PhD in biostatistics, and so we're really lucky to have him on the data science team. Some of the reasons that we called on Kurt specifically to build this interrogator prototype were because of the educated way that he could go through as an end user and really evaluate, as he's building these tools, evaluate whether or not it's a kind of tool that he might like to use if he were coming to the platform as an end user. So we had a lot of conversation with Kurt, with our quantitative medicine team, our chief science officer, with some of the scientific expertise from the Friedreich's Ataxia community, and all of that went into this prototyping process. So I'm just going to start by framing the conversation to say that although this prototype is built in our Shiny and the data subject are Friedreich's Ataxia data, the idea really from Kurt's perspective and mine was that this was really a discovery process about really eliciting requirements for the kinds of tools that we'd use for data exploration. And so our work used Friedreich's Ataxia as an example and an R Shiny app as an example for the prototyping exercise. But I don't want anyone to have the impression that we're limited to that disease or that particular tool. So we're going to talk about that a little bit more broadly as I wrap up my section of this discussion. But I can see Kurt. Hey, Kurt.
Hi.
I can see that we're jumped right into the app, right? So up at the top, I see a blue bar, and we're highlighted on the Domain Explorer. So I recognize SDTM, and for our users who are joining us, I think that some of what we're talking about is SDTM, which is a standard from CDISC. So folks who are familiar with the medical, the pharmaceutical procedures, regulatory processes will recognize SDTM. But what I'm kind of wondering here, Kurt, is do I have the ability to explore other domains within SDTM? Because I'm looking at demographics now.
Yes, you do. There is it's not shown in this picture, but there is actually a sidebar, and that sidebar has all of the different domains that are available within Friedreich's Ataxia, and the default that we start with is demographics.
Which makes sense. Everybody has a demographic. Every study has a demographics domain. Okay, Vicki, we can jump to the next slide then. I think the next thing that we're looking at is probably the next tab. Yep. So we're going over to visualizations. So Kurt, give me a tour. I'm looking as a new user coming to this platform. I'm noticing a couple of interesting things. I can choose particular variables, and over on the top right, I have univariate, bivariate, and stats. What are we looking at here?
Yeah. So, and also right above those, you'll see a data filter.
Okay.
And so you can filter the data, and whatever variables are in your domain, you can filter on those. And those filters will be applied to both the plots and the statistical variables.
So am I able to see, Kurt, I'm guessing these are not the SDTM variable names. These look like more human readable names for these variables, but am I able to look and filter more than one domain at a time?
Yeah, so, and again, this isn't shown on the screenshots, but yeah, there is an option on the side. And so all of the domains, except for the one that you picked on the first page, will be shown. And you can join 2 domains.
I see. Oh, that's really helpful. That's actually been a challenge. That's been a challenge back when I was doing some basic analyses, being able to connect one SDTM domain to another. That's frustrating for me as an analyst, as a data scientist in the past, because oftentimes you'll see these domains and they live completely separately. So that's a nice feature. Okay, Vicki, next tab. So we're starting with univariate stats, right?
Yep. Yeah. So, and you'll see that the variables that are pulled up there are again the ones from your domain, and the first the first variable or the first plots are univariate,
Right.
and so that's just one variable, and you can choose either continuous or categorical.
Gotcha.
Yep.
Oh, and then it makes assumptions for you based on. Okay, great. Let's jump to the next tab there, Vicki. Okay, we're still on univariate. So this, I see histograms, I see box and whisker plots, but Kurt, what I've noticed is you've selected the variables there. You have age by sex. Did you have to write code in order to get these histograms or box plots, or is this just a feature of the app?
That is a feature of the app. And so if I picked a categorical variable, then you would end up getting box plots. In this case, we picked a continuous variable, and so you get a histogram.
As well.
And you'll with continuous, you'll get box plots, and it separates. So you have the grouping variable there by sex. And so your group, you can see that in the histogram, male and female. So yeah, so your separate box plots, and so you can analyze the distribution here of these variables.
So as a person who has done a lot of stats modeling, I'm guessing you're going to start looking at the distribution for qualities. Vicki, let's go to the next slide. Yeah, so we're exploring bivariate, right? So tell me what's different about this exploration or visualization.
Yeah, so bivariate is two variables, and you can choose one continuous, one categorical. In this case, we pick two continuous variables, and you'll get a scatter plot. And so you'll see there, when you pick two continuous, you'll actually get the option in the upper left corner there, and it says low S.
Right.
And you can choose if you want a low S line or not. So you can actually choose those.
I'm noticing I could also choose, it looks like, can I have both at the same time, the low S line as well as the 95% standard error line?
Yep. Yeah,
Okay.
so, and it's user, I don't want to assume anything, right? So we, some people just want low S, maybe some people want both. So I offer both.
That's great. Okay, so next slide, Vicki. Yep, I knew we were getting here, not only because I've seen the slides before. This is just logically the next step that I take if I were exploring data. So I'm seeing this orange, what looks very much like just a normal distribution for comparison, right? Walk me through this, Kurt. And we're on the stats tab at this point, looking at normality.
Yep. Yeah, so we picked the statistics tab, and that is, the next logical one, because, we've already looked at, distributions and stuff. And so here we actually want to choose, is there a difference of this dependent variable between these groups is what we are trying to analyze. And so the first step of this is to do normality. And you can see there is a Shapiro-Wilks test, and that is actually just a test that's done, but also I do plots. And the reason I do plots is because plots are worth 1,000 words, right? And so the tests aren't necessarily accurate. And so I like looking at these two plots. You're right, this is the normality overlay over the histograms, and then you'll see right below that is the QQ norm plot. And if that is a straight line, then it's normal. In this case, we look at the Shapiro-Wilks, and it's actually 0. And so it's showing that it's not normal. But to my eyes, that looks pretty
normal.
Yeah, it looks pretty good to me. So that's why we have the test.
Working with life science data, if you're a stats person who hasn't worked with life science data, it probably makes you uncomfortable, but you almost never get a perfectly normal distribution. I mean, life science data are messy, right? So your eyeballs are probably a good test sometimes, especially with experience. Vicki, let's go to the next slide. Okay, so we're still on the stats tab, but now we've moved over to looking at variance. Give us a tour of the variance checks.
Yeah. So this is the same idea as normality. But in here I have the Levine's test, and I have box plots. And so I just want to look at the spread of the two over the groups. And if you look at box plots, they are a good plot to be able to show what the variance looks like. And that does look very homogeneous to me. And actually, this is a case where the Levine's test agrees with me that it is. So we get both, we can look at these plots and do the Levine's test. So, yeah.
Right. Okay. So basically, you're providing some canned options for basic testing the assumption of normality and testing the assumption of equal variance.
Yeah.
Next slide, Vicki. So now we've gone through both the normality and variance checks. I'm starting to see options for measuring difference between groups, right? And there are different tests that you might use based on the results of the normality and variance checks. We talked about, Kurt, that the normality assumption failed the test, but we thought our eyes thought that it looked normal. Am I able to choose a test even if I disagree with the results of the normality check?
Yeah, so you'll see here that there are 3 different tests.
Okay.
And so what happens is through the app, I checked the Shapiro and I say, all right, what is the p-value of that? If it's below 0.05, it failed. So if then I do homogeneous Levine's test, and if it is, bigger than 0.05, then, pass it. So then those are the 2 assumptions that I do, and I go and I find what the default test would be, right? If those were correct. And so you can see there that this is, you can also hover over these tests and you can see what the assumptions are. And so for that one that it selected, it's assuming not normal, but homogeneous variance. And so you can, as a user, I wanted to make sure that you could change those because you can, like this is showing, it is normal and homogeneous. So yeah, I'd actually not do the test that it defaults to. You can change it.
Okay. Next slide, Vicki, please. So yeah, you've just selected a different radio button, right? So you're able to proceed with your test.
Yep.
Before we talk more about the results of the student's t-test, I'm noticing something that I've overlooked to mention in the bottom left-hand corner, and I see missing. Can you talk to me a little bit about what those options are, whether I can change them, and what it actually means?
Yeah, so on those, there is potentially missing variables in these data. And so sometimes people may actually want those. And so when the app finds that those are missing, it gives you the choice of, do you want to remove these? And actually the default is yes, because usually people do, but you may not. So because in the app, I don't want to just do things in the background. I would actually want to make it so that people choose what options that they want to do.
Yeah, that's great. I can imagine a scenario where you do the student's t-test with the missing variable and without, by excluding it, just to take a look at how they compare. For example. Yep.
Yeah.
All right, thanks a lot, Kurt, for the tour of the Friedreich's Ataxia R-Shiny app. Great work. I'm going to come back and maybe you can join me at the end of this conversation because I'm going to close by showing the app that you've created here integrated into our new platform. So thank you.
Okay.
So for kind of resetting and calibrating how this discussion about statistics fits into the larger discussion about Rare Disease Cures Accelerator, I think that I described that we, and Vicki also described that we did this as a prototype development specifically, not to learn about Friedreich's ataxia necessarily, or not to perfect a shiny app. It was really about creating a prototype so that we could walk through the challenges that we would find in our new platform before the platform itself was available for, refining and tweaking. And so some of what we have learned, and the first point here, we added a bullet in italics to just let everyone know that this particular R-Shiny app has already been integrated into our new platform. I'm going to circle back, like I said, and talk with Kurt about that later. But the key learning that we really extracted from this process, more than the app, is that data should be easy to find and use, right? And so making visualizations that facilitate a lot of the kinds of deep exploration that you might do before you want to access data for a particular model or statistical tool is so important. Being able to quickly modify the views and apply different tools are important as well, because what we didn't want is for a user to have to come to our platform and do some of this work, but have to write the R script involved with producing a basic histogram. I mean, there are definitely some basic steps for exploring data as a quantitative user, for example. There's some basic steps that everyone will sort of walk through. And so why have everyone write a bespoke script in order to get some of that visualization? And then finally, the high level data visualization should be available prior to the data access request. And that's going to take me into a whole additional topic. But we've all experienced going to a data platform that has data that seem intriguing. And there's this process of saying, I'd like to have access to the data, but in order to get approved as a user of those data, I have to provide a stats analysis plan. And sometimes there's this frustrating process of, I'm not sure exactly what I would do with my stats analysis unless I knew exactly what the variables looked like and what kinds of measures you have in the data. And so there's this ridiculous catch-22. And I'll tell you honestly that in the legacy platform at c-path was a bit of the case as well. So people would require access to it, but simply download the data and start finding out if it was going to be useful for their analysis. What we want to do with the RDCA-DAP is to bypass that whole frustrating process, make it more efficient, give people the ability to do some basic exploration to say, yes, this particular study will be valuable to my analysis. This particular study is worth going through the process of writing a stats analysis plan and requesting access. Or I'm going to keep looking because this particular data set is not actually helpful. So, this led us, our learning from this prototype development process, it led us into thinking much more heavily about metadata itself. In the legacy platform at c-path, we would make metadata in a very simplified and summarized way available on different disease consortia, their websites. And we would have metadata that we derived as data managers, for example, that we could walk through users in a manual way. But the focus in the RDCA-DAP is much more about creating access to metadata from the get-go. So we've been modifying our data access agreements our data sharing agreements so that we are able to provide summarized metadata for our data sets and allow users to immediately explore what data are available, some of the metadata about those studies so that they can make that decision about whether or not it's worth their time investment to explore the data more deeply. And so what you're looking at right here is a view of our new platform. In our Fair Services catalog, we're doing a basic search. You see here that I'm searching on the phrase broken bones, and I find right away Duchenne muscular dystrophy study, a particular study where broken bones are a key word. And I think next, Vicki, yep. So another example is that I'm searching on ataxia, and if I have other types of ataxias, they'll all be included here, but you're seeing some Friedreich's ataxia data pop up based on their keywords. This is all part of the metadata catalog that's included in our new platform. I'm particularly excited about it. It might not seem like an obvious topic to go from the prototype development with basic biostatistics and data exploration. It might not seem logical to jump into metadata, but what we really found is that the umbrella topic here is making data more findable, accessible, interoperable, and reusable, and that must involve metadata as part of the exploration process. So here you'll see on a different tab, we're focusing in on a particular study. So let's say I've done a basic search in the metadata catalog and I found a particular study of interest. You can see all of the metadata variables that are provided as part of the sharing of the data in the Fair Services catalog. One of the things that I'm particularly proud of is that with the name of the study below, you see a one-line description. At the end of that description, please take note of the fact that the curation level is indicated as full. So what we have found is that with the Rare Disease Cures Accelerator data and analytics platform, we're expecting to get lots of disparate data from lots of different sources and also from different formats, from registries, from clinical trials, and all different kinds of data. And that means we needed a way to make sure that we're doing a really responsive curation process and sometimes an iterative curation process. So if we have many data sets, it is my team's directive that they get those data sets and those metadata uploaded to the platform as quickly as possible to make it accessible to everyone for finding. And these are the metadata that are going to be included whenever that study is uploaded. What you see here, though, is that this has full curation. And what that means is that in addition to the metadata and the actual physical access to the data, there's been a whole cleaning and standardization set of rules applied to these data. So they've been filtered and cleaned up and standardized in a way that will make them much easier to use. Okay, next slide. So here, we're talking about this as a topic, metadata. It's more than just a description of the study, right? One of the things that has helped me as a data scientist is to really take a deep look, and I have a strong background in database theory, for example. So I'm always eager to look right at a data dictionary. And so this has added a little bit of work on the front end of our data management process, loading not just metadata that describes the study, but also a full data dictionary that goes along with the study. And these detailed data dictionaries are going to be available for all the published data that have the minimal, the basic curation level applied to them. And you can see some examples. I mean, some of the things as an analyst that I might want to know are whether or not I've got quantitative measures. Are they numbers or categorical variables? Do I have continuous or categorical variables? What sorts of the names of the variables sometimes are not deeply explanatory, right? So having a description field that tells me exactly what the content of that data, that's helpful. And the reason that that's helpful, I will tell you as a user, is that having a list of variables that are available sometimes gives you the false hope that you're going to get some sort of a quantitative measure when it actually turns out to be just a set of free text, for example. So having some context, I don't yet have permission to fully access this patient level data set, but I have this data dictionary to give me a good description of what I can expect to find in each of the variables. Okay, next slide, Vicki. Yeah, so perfect, a little example here for this particular field. This is all the bit of information that I'm going to find that's available. A good description, as well as names. Okay, next slide. So now we've talked about metadata as metadata and data dictionaries and curation levels and all the things that would make a user who comes to RDCA-DAP able to be empowered to really not only find and access data, but also to do some visualizations and some exploration, right? And so what we learned is that the R Shiny app which I will circle back to, is one way to get some basic visualizations that we expect. They can be particularly honed into particular therapeutic areas, for example, especially in the cases where we're using data that have been mapped to an SDTM format. In this example, though, on this slide, what you see is that that process also helped us to select the base platform from a large pool of potential vendors based on some of those visualization tools, right? So this new platform that we're launching in September, it has some basic built-in analytics tools that will look very similar to the RShiny app. We have the ability to access some of these data and do some basic stats, or a user can write an R script and customize the visualizations as they wish. But this particular bar chart that you're seeing is just a built-in biomedical stats tool that's part of the platform. So that means that some basic visualizations are available for all of the data sets. Okay, next slide there. So in kind of wrapping up this topic in order to kick off the Q&A session and make sure we have a robust conversation and dialogue, I want to just wrap it up and bring Kurt back into the conversation because all of that work that Kurt did in building the RShiny app and refining it led us to a really robust list of requirements and usability features that we were looking for from data visualizations and exploration. But nonetheless, Kurt also perfected the particular Friedreich's Ataxia app. And what we're showing you here on this screen is that the Friedreich's Ataxia app that he showed you has been fully integrated into the new platform, so that when we have a production release of the platform, any user would be able to, if you have permission to access these underlying Friedreich's Ataxia studies, then you would be able to use the Friedreich's Ataxia app that we've been walking you through here. But more than that, I think that the really nice feature of our platform is that we can integrate any RShiny app. And so this has offered us at c-path an opportunity to really showcase and make more available all of the fantastic and groundbreaking quantitative models that our quantitative medicine team will produce. So many times, Kurt, give me one other example. I think maybe you've worked in Duchenne's muscular dystrophy as well.
Yeah, there is a DMD app that that we will put up. It's it's in the process, in the pipeline to be put up there
Into the platform.
Yeah. Yes.
Okay. So the reason that that's worth mentioning, I think, is that what we're expecting to offer for RDCA-DAP is not limited to data. We're also offering tools, obviously, and additionally, we're offering models that have been put through a rigorous regulatory endorsement process. And so some of our quantitative medicine efforts have led to disease progression models or clinical trial simulators. And many times we've struggled in the past at c-path to be able to share those in a standardized way. And one of the complaints that I've heard from Klaus Romero, our chief science officer, is that we have this fantastic model, but it has these really specific R packages that need to be downloaded. And so there's a lot of user support to get a person to be able to physically download the model, to apply it to their data and so forth. This bypasses all of that, right? So if we put an RShiny front end on a model that has received regulatory endorsement, it might be useful for a particular therapeutic area. We can integrate it right into this app without having to go back to the developer and pay for custom coding. Am I correct in all of that assessment?
Yeah. Yep.
So we're excited that don't you think as a statistician, it's exciting to have a place to just have a home for these kinds of apps and models and efforts? I think in the past, I might have done a lot of work and then it sort of gets dusty in a closet because there's no public sharing of a particular model, for example, right? So I think this is a great feature. And with that, I think our next slide is opening. Yep, acknowledging our FDA funding and then really just opening the floor to all of our panelists. Alex, if you would like to join us, and Vicki. And I hope we've been collecting lots of good questions in the chat from all of our participants.
Thanks everybody for the great presentation. We have some great questions coming up from the chat. So the first one, I think, is a two-in-one questions coming from Erica and Ria. Erica wanted to know what is the data source of the examples that you showed in Friedreich, but she also wanted to know, can you get access to the curated data on the platform? And Ria wanted more details on the creation process, and if you could explain how does the process differ between different sources of data that we're getting?
Oh, that's a great question. So a lot of, I think that I failed to mention something, Alex, that I think is really important, which is that a part of our curation process, as I mentioned, it's becoming more iterative. And then the other thing that I think is worth mentioning is that we're adding ontologies and semantic concepts, annotation, in order to make the data more findable. But the process in the past was quite simple. We would take in clinical trials data, and in some cases, natural history studies or other clinical studies, and we would map them to the appropriate SDTM domains for that therapeutic area. That's changed very dramatically. So the first part of my answer will be that, and Kurt probably knows better than I do about the source of the Friedreich's ataxia data, but we have some legacy Friedreich's ataxia studies that have been aggregated across, so there's not a single answer, right? It's an entire database of Friedreich's ataxia data from multiple studies that have been aggregated into the SDTM domains. So the demographics domain will have patients from several different studies, clinical trials, as well as observational studies. And then the second part of the answer specific to Friedreich's ataxia is that if you have access to this platform and you can find the data and make a request, then you can absolutely have access to the data. It will be available to qualified researchers who submit a request and meet the requirements for being a researcher in Friedreich's ataxia. Then secondly, the process itself. I'm going to stop monologuing and let other people talk, but I do think that's a really good topic, which is that the process itself has gotten much more complex in response to the complexity of having to have many different paradigms of data combined with many different disease areas. So that's particularly challenging for rare diseases, right? And none of this works if we're not able to combine the data in a meaningful way. And that's where our new pipeline ingests data using a common semantic model as opposed to a physical format, so that we're not mapping them all directly to SDTM. And in some cases, for registries, for example, there isn't always a good fit. And so we're kind of putting a square peg in a round hole. So what we're doing now is ingesting data into a common semantic model, and we're writing tools that can translate from that common model into SDTM upon export. So you could request the data in SDTM format, or you could request the data in some other format, and I'm drawing a blank. FHIR standards, OMOP standards, SDTM standards, lots of different ways that data can be shared, right? But particularly important in that process is that data quickly gets shared with metadata, and that's different from the past because we didn't share it until it was fully curated. We're making data accessible if a user would like to, before we have the chance to finish the full curation process, perhaps they'd like to take the raw data and work with it themselves and do some curation and make some of those changes. And then we have, based on how many requests for access, we have a process where we're calling it responsive curation, where we can put more data management effort into doing the full cleaning. And then the final stage of curation is annotation of ontological terms, which mean, just very briefly, ontologies. So terms that give you an idea of what the actual meaning of a variable is, to make it more findable. So if I were to search on an ontology term, something that has to do with locomotor function, that might have been annotated across many different therapeutic areas where that's a concern. And I would find all of those studies that have that ontological term annotated. And then in closing, I promise, I think that it's worth mentioning that we have on our c-path YouTube channel, Kurt has recorded a video where he gives you a really detailed demonstration of the Friedreich's ataxia app. Because we're not having a live full production release of the platform until September, this gives you a good sneak preview at what the platform itself looks and feels like. And then the second webinar I would point you to is that Dr. Ramona Walls from the data science team has recorded a webinar that explains the basic concept of ontologies, but also why it's helpful in the rare disease space.
Well, thank you, Amanda. I have another question. I think it relates to the added value of the platform. So ID was wondering, for more savvy researcher, can you touch base on what the added value of this platform? ID also wanted to know if there would be access of the raw data for savvy researchers again that want to do their own coding and analysis.
Yes. So we haven't really mentioned this because we focus so heavily on exploration in the FAIR services catalog, but I think of our new platform as sort of a phased approach to data access, because you can do some searching of metadata and basic exploration. But then once you've honed in on the particular studies that you'd like to access and you make a request for those, you can move them into your own digital research environment or workspace. Once you've moved the data into your workspace, you can work with the raw data. You can do whatever sort of standardization or curation you'd like to do. You can sort and filter. You'll have access to Python. You'll have access to R. You'll have access to some other proprietary tools that we use in our quantitative medicine team, for example. So it's basically, Alex, as if you're able to take the data that you've decided that you want to invest your time in, and you're able to put them into your own private computer to do analysis. The other great feature of this workspaces is that if you and I were working on a collaboration together, and let's assume that you're at a university and I'm here at c-path, I can invite you to my workspace and we can work together and see the same data and work on the code together as well. Do you have something to add, Kurt or Vicki, to that process?
Nope.
So I think the second part of your question, maybe I'll open it up to Kurt and Vicki for this too. You, Alex, you asked, why does this particular platform add value to the rare disease space? And I think, unless you guys want to take it, I will jump in with ontologies again. We're investing.
I think if you want to jump in with ontologies as the value add, that's a great point. We've got some additional follow-up questions on ontologies, so let's go back to that.
Okay, great. So I think what makes this platform quite different from any other platform that we've developed at c-path, but also different from, honestly, the platforms we've collaborated with, data platforms all over the world. There are a couple of things that make this quite different and much more ambitious. Number one is that access, as I mentioned, to a really robust set of metadata and data dictionaries so that you might be able to access data in a more thoughtful way. But the second is that c-path, in my experience, and I've been very lucky to access some incredible platforms around the world, but in my experience, c-path spends a lot more energy on aggregation of data. So in the past, that was physical aggregation of data. But for Rare Disease Cures Accelerator, I think it's particularly important to talk about the value of aggregating data in ways that weren't predefined. And by that, I mean that if I'm a researcher and I'm studying a disease that's so rare that there really aren't any sort of rigorously collected scientific data in my therapeutic area, I might at least be able to search on other diseases that have a similar symptom and what those, whether or not there have been effective therapeutics for a particular symptom, for example. I'm able to aggregate data without having, so being disease agnostic, honestly, having the ability to annotate with ontologies helps us to find data that have a similar meaning, even if the variable names are completely different and not obvious to a person, right? So in the past, we would guess at well, if you're an Alzheimer's researcher, you'd like to have the data from all these studies aggregated into one single database and kind of lumped together in that way. And that aggregation was incredibly valuable. But what we're attempting to do with RDCA-DAP, and I think where we're being successful, is that in the next year, we'll be rolling in these semantic tools in such a way that it won't matter if you're looking at a particular feature of a particular disease, you'll actually find all of the studies that have included some sort of measure related to that particular disease feature, even if the diseases are completely unrelated in their origin.
So Amanda, a couple of the follow-up questions specific to ontologies that we had, and I'll read the questions, but they were forwarded, so I apologize for not knowing exactly who's answered them. I'm wondering whether the hierarchies of disease and phenotype ontologies are being leveraged to A, improve search results, or B, use the hierarchies somehow in statistical analysis. And then a follow-on to that, I'll give both of them to you and then you can address which disease ontologies are being leveraged in particular, and what do you do if you need to code a disease that is not present within the ontologies utilized? So really great questions around our ontology development, and truly an area of a great amount of focus for us and a lot of effort going into this to make sure we're getting that right. So maybe talk a little bit more about some of the great work that Ramona and her team are doing around that.
It's a great way to sort of round out the conversation, I think. So Alex, is there something else in addition to what Vicki just mentioned? Do we need to make room at the end of the conversation for some other topic, or shall I really dive into that one?
I think you should dive in. I mean, if you have time, one of the questions, I think somebody wanted to have some examples of success that we had analyzing some of the rare disease data that we have, you know, if not in the program, you know, other rare disease consortiums. But just some kind of example of the power and what we achieved so far.
I think I'm going to do a terrible job of summarizing it just off the cuff, but I think what we can do, Alex, is follow up with an email to our participants that lists some of our regulatory successes. I think we have them. We've worked in polycystic kidney disease, and that's probably my favorite one to brag about, but I don't want to be biased. We've worked in Duchenne muscular dystrophy, Friedreich's ataxia, polycystic kidney disease, Huntington's disease. So we've had lots of successes from combining data. But I will tell you, we haven't had successes from the Rare Disease Cures Accelerator platform specifically because we're in the beta testing phase, right? And we'll be going live in September. So I think we have a good track record that will continue to do that. But I think that Vicki's ultimate question kind of ties into this as well, which is, you know, about ontologies. I'm going to answer that in part, but I'm also going to refer people again to the webinar that Ramona's put together, and also to ask for some standardized responses from Ramona that give answers to these questions in the chat as part of an email, because I want to make sure that we really do these answers justice, and I'm going to just summarize. But I'll tell you a couple of things. One is that we have been working on partnerships with other folks around the world who are far more experienced with ontologies. And I think anybody who knows what that term means, if you've worked in genomics or bioinformatics, you realize that they have a huge head start in molecular biology with the use of ontologies and semantic terms and searches. So we've been partnering with folks to develop these relationships. Dr. Ramona Walls and the data science team is leading this effort and really architecting these solutions. And she's part of boards, she's part of working groups. She's been doing this for many years in an academic setting. So we're developing partnerships for two reasons. One is that we're working on a pilot with the European Joint Program on Rare Diseases that will specifically explore some of the challenges of using a common semantic model and then do building searches based on ontological terms. So we're doing some road testing using polycystic kidney disease as the example data from this partner as well. So sharing some of our tools and some of our lessons learned, right? But the other thing that we're going to be doing is when there is not an appropriate ontology term, we will be working with the owner. We're working already actually with the OVO Foundry, and if you I'm sure that people know them. They're providing some very customized training to our internal staff that they will then pivot to providing training to everyone in the rare disease community who would like to learn more about ontology and annotation, and specifically probably on searching based on ontologies, right? So when there is not a good coverage of a particular disease or symptom, we'll be working to develop new ontological terms. It will be an active engagement from our team. And then the other thing, the question about what disease ontologies are we using? I'll tell you that that's an open question. So we are really shifting our focus away from the basic platform functionality and requirements elicitation, so that in the next year, after we've launched the base platform in September, our team will be spending the next year heads down integrating ontologies and building a graph database and a search index that will allow us to use semantic web tools to be able to find these data sets based on their semantic meanings, right? So stay tuned, because we have huge plans for that, investing in that particular area. But those were great questions about ontologies, and I'm eager to make sure, Alex, that we follow up with an email that points people to those webinars and to some of Ramona's more detailed answers for everyone.
Right, well, thank you for your answer, and thanks everybody for the great presentations and the great questions. Don't forget to follow our next webinar series, which will be on each Wednesday of May and June. We will provide you with a link to this webinar. And next slide, please. Just to conclude, the rare disease platform work for rare disease never stops. We're always acquiring new data. So if you have further questions on the program, or if you want to contact me to talk about data sharing with us, you can reach out at the email on this slide. Again, thanks everybody for joining today, and have a great day.
Thanks, everyone. Thanks for your time.